Yeah, in previous class we completed nucleus. Imagine this is the nucleus. So in previous class we completed internal nucleus. The thread-like structures which are present in nucleoplasm, the thread-like structures are called as chromatin. Mind. Time pass. Chromatin material undergoes condensation, results it forms what? Chromosome. So chromosomes information we are going to discuss now. So generally chromatin material undergoes condensation, result they forms the chromosome. See, in the structure of chromosome center one spherical body present. See, this is the chromosome. In the structure of chromosome center one spherical body present. That spherical body is called as centromere, which is also called as primary constriction also. Center one spherical body present, that spherical body is called as centromere. The centromere is also called as primary constriction. And see, both sides of centromere, of disc-like structures are present. These are called as kinetochores. The off disc-like structures are present both sides of centromere. These disc-like structures are called as kinetochores. How many kinetochores are present? 2 kinetochores. How many centromeres? 1. And see, in the structure of chromosome, 2 arm-like structures. This is one arm-like structure. This is the another arm-like structure. These arm-like structures are called as chromatids. This is one chromatid and this is one chromatid. The 2 arm-like structures present both sides of centromere is called as chromatid. How many chromatid? 2 chromatid. In every chromatid, one DNA molecule. See, this is one DNA and this is one DNA. Every chromatid consists of one S DNA. And see, whatever structure present, either above chromatid or below chromatid, it is called as secondary constriction. Just above the, see, not in all chromatid, in some chromosome, see, not in all chromosome, in some chromosome, just above the secondary constriction, one more spherical body present, it is called as satellite. That satellite not present in all chromosomes. Just above the chromatid, one tube-like structure present, it is called as secondary constriction. Just above the secondary constriction, one more spherical body present, it is called as satellite. Satellite not present in all chromosomes. Satellite containing chromosomes are called as SAT chromosomes. జనరల్గా శాటిలైట్ అన్నిట్లలో ఉండదు. సో satellite containing chromosomes are called as sat chromosome. Clear? Come. See, listen. One chromosome equals to how many centromeres? One centromere. Ante kada na, only one centromere. And one chromosome equal to how many kinetochores? Two kinetochores. This is one, this is one. And one chromosome equals to how many chromatids? Two chromatids. Okay. See, to study the structure of chromosome. See, this is the structure of chromosome. To study the structure of chromosome, metaphase in cell division, metaphase is the best phase. Metaphase is the best phase to study the structure of a chromosome. Very important. Prophase, metaphase, anaphase and telophase. 4 subphases are present in cell division. After this lesson we will discuss. So to study the structure of chromosome, which phase is the best phase sir ante metaphase. Sir, why? Why metaphase is the best phase to study the structure of chromosome anante, during metaphase chromosome condensed maximum. So chromosome condensed maximum, that's why metaphase is the best phase to study the. When maximum condensed, that chromosomes are visible very clearly. Maximum condensation అయినప్పుడు ఏమవుతుందన్న, maximum condensation అయినప్పుడు ఏమవుతుంది, that material visible very clearly. That's why metaphase is the best phase to study the structure of chromosome. See, on the basis of ఇదేంటిది నాన్న, this is the centromere, correct? అంతే కదా నాన్న, this is the centromere. On the basis of centromere position, on the basis of what? On the basis of centromere position, chromosomes are classified into 4 types. On the basis of chromosome position, on the basis of centromere position, chromosomes are classified into 4 types, which include, first one is the metacentric chromosome, second one is the submetacentric chromosome, third acrocentric chromosome, fourth telocentric chromosome. So on the basis of centromere position, chromosomes are classified into 4 types: metacentric, submetacentric, acrocentric and telocentric. Metacentric sir ante, meta ante center. See, centromere present in the center of chromosome. This is the centromere. Centromere is also called as primary constriction, correct? See, centromere present in the center of chromosome, that chromosomes are called as metacentric. Here centromere present in the center, that's why 2 chromatids. See, this is one chromatid, this is the another chromatid, both present in equal size. ఎందుకంటే centromere present in the center, that's why what happened nana, that's why what happened in nana, it is present, 2 chromatids are equal in nana size or length. Submetacentric means, see, centromere present just above the center. This is the centromere. The centromere present just above the center. That's why one short chromatid, one long chromatid is developed. It is called as submetacentric. And acrocentric means centromere present near to one end. Centromere present near to one end. This is the centromere. ఇది ఎండ్కు నియర్గా ఉంటుంది. That's why one very short chromatid, one very long chromatid is there. That type of chromosomes are called as acrocentric. Telocentric means centromere completely present end, nana. This is the centromere. The centromere present completely end, it is called as telocentric. Clear? What is metacentric, nana? Centromere present in the center of chromosome. What is submetacentric? Centromere present just above the center. Acrocentric, centromere near to end. Centromere completely end. Telo ante end. And metacentric chromosomes are appeared in V shape, submetacentric L shape, acrocentric J shape, telocentric I shape. Means chromosomes are appeared in 4 shape: V, L, J, I. So V shape lo undedi metacentric, L lo undedi submetacentric, acrocentric J, telocentric I. Clear? Is it clear? Any doubts? Any doubts? Yeah. So this is the centromere related. After the centromere, we are taking the cilia and flagella. So cilia and flagella both are cilia and flagella both are locomotory organ, means it provides the movement to the organism. Internally cilia and cilia and flagella in similar in structure. Internally, cilia and flagella consist of 2 components. First one is the protoplasmic membrane untundi. Second one is the axonema or axoneme. ఈ ప్రోటోప్లాస్మిక్ మాస్ మనకు అవసరం లేదు, యాక్సోనిమ్ స్ట్రక్చర్ మనం చదవాలి. Cilia and flagella both are the locomotory organ. Internally both are similar in structure. Internally cilia and flagella consist of internally cilia and flagella consist of 2 components. First one is the protoplasmic membrane, second one is axonema. This axonema is very important, then only we go for the protoplasmic membrane. Axonema internally it shows 9+2 arrangement. So axonema internally it shows 9+2 arrangement. 9+2 arrangement ఎలా ఉంటుంది అంటే ఇక్కడ చూడండి, I am explaining axonema only. So in case of axonema structure, center 2 microtubules present. This is one microtubule, this is the another microtubule. This is the C1, C2. Center how many microtubules present? 2 microtubules, C1 and C2. Little bit big size చేస్తున్నాను. Okay. So internally 9+2 arrangement is there. In 9+2 arrangement, 2 microtubules present in the center, they named as C1 and C2. This C1 is made up of, C1 made up of 10 protofilaments, nana. 10 protofilaments are present. In C2, 13 protofilaments are present. Protofilament ante threads made up of protein. In case of C1, 10 protofilaments are present. And in C2, 13 protofilaments are present. In C1, 10 protofilament, in C2, 10 protofilaments are present. This C1 and C2 covered with one sheath, it is called as central sheath. Labeling తర్వాత ఇస్తాను. ఇది 9+2లో 2కు సంబంధించి. నెక్స్ట్ పెరిఫెరీ కన్సిస్ట్ ఆఫ్, periphery consists of 9 microtubules. 1, 2, 3, 4, 5, 6, 7, 8, 9. So internally 9+2 arrangement is there. సెంటర్లో 2 ఉంటాయి, పెరిఫెరీలో ఎన్ని ఉంటాయి? 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. సో పెరిఫెరీ 9 ఉన్నాయి, సెంటర్ 2 ఉన్నాయి, కాబట్టి దట్ అరేంజ్మెంట్ ఈజ్ కాల్డ్ యాజ్ 9+2 అరేంజ్మెంట్. బట్ సి, పెరిఫెరీ మైక్రోట్యూబుల్ ప్రజెంట్ ఇన్ డబ్లెట్ ఫామ్. ఇగో ఇలా ఉంటాయి. పెరిఫెరీ మైక్రోట్యూబుల్ ప్రజెంట్ ఇన్ డబ్లెట్. సో అందులో ఒకదాన్ని ఏ అంటాము, ఒకదాన్ని బి అంటాము. దిస్ ఇస్ ద ఏ అండ్ బి, ఏబి, ఏబి. లోపల ఇస్తున్నాను లేదు. సో పెరిఫెరీ మైక్రోట్యూబుల్ ప్రజెంట్ ఇన్ డబ్లెట్ సి, ఏ బి. ఏ ప్రజెంట్ స్లైట్లీ ఇన్సైడ్, బి ప్రజెంట్ స్లైట్లీ అవుట్సైడ్. Periphery 9 microtubules present. Periphery 9 microtubules present. In that, A microtubule present slightly inside, B microtubule present slightly outside. And see, A microtubule is connected with another B microtubule. See, A of one microtubule, A of one microtubule connected with B of another microtubule in cyclic manner. Ego, A of one microtubule connected with B of another, A of microtubule connected with B of another microtubule, A connected with B, 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 A connected with B. A of one microtubule connected with B of another microtubule, it is called as AB linker అనే పేరుతో పిలుస్తున్నాం. And see, listen, from every A microtubule, one spoke-like structure grows towards the center. From every A microtubule, one spoke-like structure grows towards the center. It is called as radial spokes. How many radial spokes are present? 9. See, 1, 2, 3, 4, 5, 6, 7, 8, 9. From every A microtubule, a spoke-like structure grows center. It is called as enana. It is called as radial spokes ga cheptam. It is called as what? Radial spokes. See, ఇగో ఇదేంటిది నాన్న, వాట్ ఈస్ దిస్? దిస్ ఈజ్ ద సెంట్రల్ షీత్ అంటాం. ఇక్కడ లేబులింగ్ ఇస్తున్నాం. దిస్ ఈజ్ ద సెంట్రల్ షీత్. దిస్ ఈజ్ ద సెంట్రల్ షీత్. సో రేడియల్ స్పోక్స్ ఆర్ నాట్ అటాచ్డ్ విత్ ఎన్న సెంట్రల్ షీత్. డిస్టెన్స్ ఉంది చూడండి. సో So total how many radial spokes are present? 9 radial spokes are present. That 9 radial spokes are not attached with. That 9 radial spokes are not attached with a central sheath. But when cilia and flagella shows movement, that time radial spokes are attached with a central sheath. Generally, radial spokes are not attached with the central sheath, but during the movement of cilia and flagella, thus radial spokes are attached with a central sheath. Is it clear? All of you. So this is the internal structure of cilia and flagella. And come to the next one is the centrosome. Centrosomes consist of centrioles. Centrioles present in animal cells only. The centriole, see, one centrosome consists of 2 centrioles. See, this is one centriole and this is the another centriole. Centrosome consists of 2 centrioles. These 2 centrioles are perpendicular to each other. This one is perpendicular to this one is perpendicular to this one. Total how many is present, Nanai? Total how many is present? Yeah. So that centrioles present in animal cell only. These centrioles participates in cell division. They participate in cell division. Centrioles are participates in cell division. Internally, centrioles same like that of cilia and flagellal, but minute differences are there. So in the internal structure of centriole, see, internal structure of centriole, center one cavity is present. This cavity is called as hub. In the structure of centrosome or centriole, center one cavity is present, it is called as hub. And periphery consists of, see, periphery consists of 9 microtubules. 1, 2, 3, 4, 5, 6, 7, 8, 9. Periphery consists of how many microtubules, Nana? 9 microtubules. Kabatti center lo enni unnai? Center nothing. Periphery consists of 9. That's why that arrangement is called as 9 plus 0. Ikkada center 2 unnai kabatti 9 plus 2. Ikkada center em ledu. C1, C2 is not there. That's why this arrangement is called as 9 plus 2 arrangement or 9 plus 0. 9 plus 0 arrangement. Center microtubules are absent. That's why it is considered as the 9 plus 0. ఇక్కడ చూడండి periphery how many present? 9. Present in doublet. Doublet means A and B అనేటువంటి ఉంది. కానీ ఇక్కడ periphery microtubules present in triplet. See, A, B, C. So periphery microtubules present in triplet form. So periphery microtubule present in triplet form. A, B, C. A, B, C. A slightly present inside and B slightly present outside. So periphery microtubules present in triplet form in centrosome present in the form of ABC, A present slightly inside, C present slightly outside. And see, listen, A of one microtubule is connected with C of another microtubule in cyclic manner like C. A connected with C, A connected with C, A connected with C, A connected with C like that. A microtubules, A of one microtubule is connected with C of another microtubule. That's why it is called as AC linker అని అంటాం. ఏకు సికు మధ్యలో ఉంది కాబట్టి దీన్ని ఏమంటాం నాన్న మనము ఏసీ లింకర్గా మనం చెప్తాం. ఓకేనా? సో దట్ ఏంటన్నా A of one microtubule connected with C of another microtubule, that's why it is called as ఏంటన్నా AC linker గా చెప్తాం. And see, listen, just below the A microtubule, one dense material is present like this. Just below the A microtubule, one dense material is present. This dense material is called as X body antam. See, these X bodies are connected with, X bodies are connected with what, Nana? It is connected with A microtubule. X body, see, this is the X body, color changes. So, X bodies are connected with, Nana? X bodies are connected with what? A microtubule. X bodies are altered with, this is X body, this is the Y body. So every X body is altered with what, Nana? Y bodies. Finally, X body and Y body, X body, Y body, X body, Y body are connected with each other. ఇలా కలిసిపోయి ఉంటాయి. Finally, X body and Y body both are ఏంటన్నా ఏమవుతాయి? X body and Y body all are ఏంటన్నా connected with each other. And see, listen, from every A body, one spoke-like structure grows towards the center. What name antam nana? From every A body, em erpadtayi nana? One spoke-like structure is developed to the center. Those are called as nana? Those are called as radial spokes. How many present? 9. Ikkada chudana. These radial spokes are finally attached with the center hub. Hub ku attach avutadi nana. Total 9 radial spokes. That 9 radial spokes are finally attached with center hub. So this is the structure of enana centrosome ga manam cheptam. So this looks like enana centrosome or centriole. It looks like one cart wheel. ఎందుకంటే ఇగో మధ్యలో అటాచ్ అవుతున్నాయి కాబట్టి ఇట్ లుక్స్ లైక్ వాట్ నానా? ఇట్ లుక్స్ లైక్ వన్ కార్ట్ వీల్. సో దిస్ ఈజ్ ద ఏంటన్నా సెంట్రోజోమ్గా మనం చెప్తున్నాం. సో దిస్ ఈజ్ ద ఏంటన్నా స్ట్రక్చర్ ఆఫ్ క్రోమోజోమ్. This is the types of chromosome on the basis of centromere position. This is the internal structure of cilia and flagella. This is the internal structure of enana centrosome or centriole. Nucleosome, last topic in this lesson. Sir, what is nucleosome, sir? Anyone, what is nucleosome? Anyone? Nucleosome means See, already I said, see, imagine this is the nucleus, correct? In nucleus, whatever transparent fluid is there, what is that? It is called as nucleoplasm అని అంటాం, అవునా? In nucleoplasm, some thread-like structures are present, correct? That thread-like structures are called as chromatin material అంటాం. See, when chromatin material observe under the microscope, when chromatin material observe under the microscope, ఇగో ఈ విధంగా కనిపిస్తున్నాను. Many means some beads like structure present on chrome. Take one chromatin, observe under the microscope, nana. Take one chromatin, observe under the microscope. Many beads like structures are present. Every bead is called as one nucleosome. This is one nucleosome, this is one nucleosome, this is one nucleosome, this is one. When chromatin material observe under the microscope, it looks like a beads on string antam, and every bead is called as one nucleosome. See, nucleosome is made up of two components, nana. Nucleosome is made up of histone proteins plus DNA. This nucleosome, nana. This bead is called as nucleosome. Generally this nucleosome made up of two component, histone proteins and DNA. Nucleosome is made up of ఏంటండి నాన్న? Histone proteins and DNA. So histone proteins point of view, four types of histone proteins are present: H2A, H2B, H3, H4. So first histone గురించి మాట్లాడతాం. Nucleosome consists of two components. First one is the histone protein and second one DNA. First I am talking with histone protein. In histone protein, four types of histone proteins are present: H2A, H2B, H3 and H4. See, you go. Imagine four types of histone proteins are present. First one is the H2A, H2B, H3, H4. So how many types of histone? Four types of histone, but each one is double. One H2A present front side, one present outside, means back side. Total how many histones ఉంటాయి? That 8 histones are collectively called as histone octamer అంటాం. Types ఎన్ని అడిగితే 4 types, H2A, H2B, H3, H4, but each one is double. So into 2 వేస్తే ఎంత అవుతుంది? 8. That 8 collectively called as histone octamer. See, this is one histone octamer. Just beside this, one more histone octamer is present. Just beside, one more histone octamer is present. This is the H2A, H2B, H3, H4. Same H2A, H2B, H3, H4. అర్థమవుతుంది? And see, listen, this is the histone octamer. These histone proteins present in positive charge. Sir, why histone proteins present in positive charge, sir? Ante because of the presence of basic amino acid, that is lysine and arginine. Generally histone proteins present in which charge, nana? Generally histone proteins present in which charge? Positive charge. Why they are present in positive charge? Because of basic amino acid lysine and arginine. Lysine and arginine ఎక్కువగా ఉంటాయి కాబట్టి they develop the positive charge to the histone protein. And next ఏంటిది నాన్న? DNA. ఇటు చూడండి. See, this is the DNA. Imagine this is the DNA. This DNA will come here and it wrapped 2 turns around the histone atomer comes out, and here also wrapped 2 turns, and next wrapped 2 turns comes out. What is this? This is the DNA. The DNA will come and wrap 2 turns around the histone atomer beautifully. Then comes out again around the histone atomer 2 turns itla tirugutu veltundi. Finally, the 2 ends of DNA, is tied by the one more histone protein, that is H1. H1 histone protein ఏం చేస్తుందన్న? H1 histone protein ఏం చేస్తుంది? It tied the 2 ends of DNA. అందుకని H1 protein మనము ప్లగ్గింగ్ ప్రోటీన్ అని కూడా అంటాం. It plugs the 2 ends of DNA. The DNA will come and wrapped how many turns? It wrapped 2 turns around the histone atomer, then comes out like that. Finally, the 2 ends of DNA is tightened by the H1 histone protein, that is called as plugging protein. See, నేను ఏం చెప్పాను? న్యూక్లియోజోమ్ అంటే ఏం చెప్పాను? హిస్టోన్ ప్రోటీన్స్ అండ్ డిఎన్ఏ. సీగో ఇక్కడ హిస్టోన్ ప్రోటీన్స్ ఉన్నాయి, దాని చుట్టూ డిఎన్ఏ. న్యూక్లియోజోమ్ అంటే గిదేనా? దిస్ ఈజ్ 1 న్యూక్లియోజోమ్, దిస్ ఈజ్ 1 న్యూక్లియోజోమ్, దిస్ ఈజ్ 1 న్యూక్లియోజోమ్. బట్ H1 ఈజ్ నాట్ ఏ పార్ట్ ఆఫ్ న్యూక్లియోజోమ్ నాన్న. న్యూక్లియోజోమ్లో ఉండేటువంటి హిస్టోన్ ప్రోటీన్స్ ఏమంటే గివే 4. In case of in the structure of nucleosome, how many types of histone proteins? అంటే 4 types. అవేంటండి? H2A, H2B, H3, H4. But H1 also histone, but it is not a part of nucleosome. It is present outside of nucleosome. So this is the nucleosome. ఇది nucleosome. ఇది ఇక్కడ ఉంది. So that's why H1 is not a part of nucleosome. In the structure of nucleosome, only 4 types of histone proteins present, that is H2A, H2B, H3 and H4. And see, DNA present in negative charge nana. ఎందుకనంటే DNA structurally consists of phosphate. Wherever phosphate is present, it develops the negative charge nana. ఫాస్ఫేట్ ఎక్కడ ఉన్నా అది నెగిటివ్ ఛార్జ్ని డెవలప్ చేస్తుందా, పాజిటివ్ ఛార్జ్ని డెవలప్ చేస్తుందా అంటే అది ఎప్పుడు నెగిటివ్ ఛార్జ్నే డెవలప్ అవుతుంది. హిస్టోన్ ప్రోటీన్స్ ఏ ఛార్జ్లో ఉన్నాయి? histone protein present in positive charge. Reason? Because of the basic amino acid lysine and arginine. And DNA present in negative charge. Reason is that because of phosphate. So phosphate negative charge DNA undi, positive charge ikkada chudana. The negatively charged DNA wrapped around the positively charged histone atomer beautifully. So this is the structure of a nucleosome. This is the structure of a nucleosome. Is it clear? What is the function of histone proteins annadu anukonanu? These histone protein makes the foldings in the DNA. ఇప్పుడు చూడండి, హ్యూమన్ డిఎన్ఏ ప్రజెంట్ ఇన్ 2 పాయింట్. ఫర్ ఎగ్జాంపుల్, దిస్ ఈజ్ ద హ్యూమన్ డిఎన్ఏ. హ్యూమన్ డిఎన్ఏ లెంత్ ఎంత ఉంటుందంటే నియర్లీ 2.2 మీటర్. నియర్లీ టర్మ్, అప్రాక్సిమేట్లీ అనే టర్మ్ చాలా ఇంపార్టెంట్. ఎగ్జాక్ట్ 2.2 మీటర్స్ అని చెప్పలే. సో ఇన్ కేస్ ఆఫ్ హ్యూమన్ డిఎన్ఏ కన్సిస్ట్ ఆఫ్ నియర్లీ 2.2 మీటర్స్. అంటే నియర్లీ 2.2 మీటర్స్ మీన్, సి, isolate the DNA of human being, unstress it. It is double to our height. మన డిఎన్ఏను స్ట్రెస్ లేకుండా చేస్తే మనతోని డబల్ ఉంటుంది. దట్ లెంతీ డిఎన్ఏ present in this lengthy DNA present in smallest nucleus, 0.1 micrometer containing nucleus లో సెటిల్ అయింది. ఎందుకనంటే అది ఫోల్డింగ్. ఇంత పెద్దది ఉన్నది ఎట్లయింది నాన్న? చిన్నదిగా అయిపోయింది. ఎందుకని? ఇగో ఇది ఇట్లా వ్రాప్ అవుతుంది, ఇట్లా వ్రాప్ అవుతుంది, టూ ఇట్లా వ్రాప్ అయిన కొద్దీ లెంత్ ఏమవుతుంది, తగ్గిపోతుంది. ఫైనల్లీ ఇట్ ఈస్ ఫిక్స్డ్ ఇన్ ద 0.1 మైక్రోమీటర్ కంటైనింగ్ ఎన్ న్యూక్లియస్. అంత చిన్న న్యూక్లియస్లో అది సెటిల్ అయింది, ఎందుకంటే బికాజ్ ఆఫ్ హిస్టోన్ ప్రోటీన్. సో హిస్టోన్ ప్రోటీన్స్ ఏం చేస్తాయంటే దిస్ హిస్టోన్ ప్రోటీన్ మేక్స్ ది ఫోల్డింగ్స్ ఇన్ ద DNA, దట్స్ వై లెంత్ ఆఫ్ DNA decreases. Finally it is settled in the, there is a possibility to settle in the 0.1 micrometer containing nucleus. And which phase is the best phase to study the structure of chromosome nana? Metaphase. అదే విధంగా న్యూక్లియస్ మనం చూసాం. టు స్టడీ ది, ఇక్కడ రాయండి నాన్న, టు స్టడీ ది స్ట్రక్చర్ ఆఫ్ న్యూక్లియస్ ఇంటర్ఫేజ్ ఈజ్ ద బెస్ట్ ఫేజ్. ఇంటర్ఫేజ్ ఈజ్ ద బెస్ట్ ఫేజ్ టు స్టడీ ది స్ట్రక్చర్ ఆఫ్ ఏంటన్నా న్యూక్లియస్గా మనం చెప్తున్నాం. అర్థమవుతుంది. అండ్ ఇంతకుముందు మనం సెంట్రియోల్ స్ట్రక్చర్ చూసాం, అక్కడ చూడండి నాన్న, centrioles are not surrounded by any membrane. That's why centrioles is example of non-membrane bound cell organelle. So generally centrioles are not surrounded by any membrane. Generally centrioles are not surrounded by any membrane. That's why centrioles are the example of non-membrane bound cell organelle. See, ఒక పాయింట్ చూడండి అన్న, లాస్ట్ సింగిల్ మెంబ్రేన్ బోన్ సెల్ ఆర్గనెల్ ఎగ్జాంపుల్ ఏంటి సార్ అని అంటే చెప్పండి. లైసోజోమ్, ఎండోప్లాస్మిక్ రెటిక్యులం, గోల్జీ కాంప్లెక్స్, వాక్యూమ్. నెక్స్ట్ డబల్ మెంబ్రేన్ బోన్ సెల్ ఆర్గనెల్ ఏంటన్నా? మైటోకాండ్రియా, క్లోరోప్లాస్ట్ ఈజ్ ద టైప్ ఆఫ్ ఏంటన్నా ప్లాస్టిడ్. ఇట్ ఈస్ ద టైప్ ఆఫ్ ప్లాస్టిడ్. ఇంకేమైనా ఉందా? న్యూక్లియస్ ఆల్సో. Nucleus is also surrounded by the two membrane, outer nuclear membrane, inner nuclear. And last, non-membrane bound cell organelles. Non-membrane bound cell organelles ఏవి అని అంటే రైబోజోమ్, centriole. Centrioles present in only animal cell. వాడు క్వశ్చన్ అడిగాడు అనుకో, ఇటు చూడన్నా, నాన్ మెంబ్రేన్ బోన్ సెల్ ఆర్గనెల్స్, నాన్ మెంబ్రేన్ బోన్ సెల్ ఆర్గనెల్ ప్రజెంట్ ఇన్ బోత్ ప్లాంట్ సెల్ అండ్ ఎనిమల్ సెల్ అని అడిగాడు అనుకో, బోత్ ప్లాంట్ సెల్ అండ్ ఎనిమల్ సెల్ అని అనుకో, అప్పుడు ఆన్సర్ ఏమవుతుంది? రైబోజోమ్. రైబోజోమ్ ప్రజెంట్ ఇన్ బోత్ ప్లాంట్ సెల్ అండ్ ఎనిమల్ సెల్. బట్ వాడు అదే క్వశ్చన్ అడిగాడు అనుకో, నాన్ మెంబ్రేన్ బోన్ సెల్ ఆర్గనెల్ ప్రజెంట్ ఇన్ ఓన్లీ ఎనిమల్ సెల్, ఆన్సర్ ఏంటిది అప్పుడు? సెంట్రియోల్ అనేటువంటిది ఆన్సర్ అవుతుంది. క్లియర్? అర్థమవుతుంది? ఆల్ ఆఫ్ ఈజ్ ఇట్ క్లియర్ ఆర్ నాట్? సో సింగిల్ మెంబ్రేన్ బాండ్ సెల్ ఆర్గనెల్, లైసోజోమ్, ఎండోప్లాస్మిక్ రెటిక్యులం, గాల్జీ అండ్ వాక్యూల్. Double membrane bond cell organelle, mitochondria, chloroplast, nucleus. Chloroplast is the example of plastid. And the non-membrane bond cell organelle example ribosome and centrioles ga manam cheptunnam. So this is the cell biology. Tomorrow we will discuss the new lesson and very important lesson, biomolecules. Any doubts you can ask me. Okay, right now.